Hello, everyone, and thanks for joining me for this first session of our RNA-seq course. So I'll just be starting by sharing the slides. So yes, so for those who don't know me, I'm Paula. I'm from cell biology, and I will be presenting the first session of our RNA-seq course together with Alistair from PNAC. And so this course is divided into two sessions. This session, which is more like a theoretical session, where I will be introducing you to some core concepts of an RNA-seq experiment and data processing. And then I will be introducing you to Pragee, which is a pipeline tool that was designed. So I designed it to help you in the data processing. And then Alistair is going to present to you PEAT, which is another software tool that he developed. And it's very user-friendly, and it will help you to look at your data once processed and ask several questions to your data, such as if you want to have a list of differential genes, differentially expressed genes, and if you want to check the log2 fold change of those genes, and if you want to compare different conditions. So that tool can help you do that through a GUI. So that is this session. Then we'll have a practical session, which was not going to be given as a webinar, but as a video, which is going to be uploaded to the LMB YouTube channel. I should also let you know, for those who are not aware, that we are recording this webinar so that we can also make it available, so that the first session and the second session are together for people to watch at a later date if they want to. Also, since this is a webinar, we're very happy to answer any questions, but perhaps the easiest way would be if you were to just write them down on the chat box, and then just once we finish the talk, we will go through them and answer them. So okay, so I think I'm ready to start, and I will start by a brief overview of an RNA-seq experiment. So RNA-seq uses deep sequencing to profile transcriptomes, and these can come from different sources, so from cell culture, from tissues such as muscle, brain, etc., or whole organisms. In this example, I put C. elegans, but you could also get it from Drosophila embryos or the different types of small animals. So you will extract the RNA. Typically it's mRNA, but it doesn't have to be. And then this RNA will be fragmented and converted to cDNA, or you can do this in a reverse order. It depends on the library type that you choose. And then once you've done that, you will go through a step of amplification, where you will ligate sequencing adapters. And at this point, you should be ready to send your library for sequencing. So it will go into a machine similar to this one, and the output will be the sequence of nucleotides from your cDNA molecules, and those are such as these, and they are called reads. So the file format that is outputted from a sequencer is a FastQ file. So that's quite standard. So once you get these FastQ files with the nucleotide sequence of your reads, usually you go through a step of alignment of those reads to the genome, so that you know where each read matches, so that you get genome coordinates. And if you're doing this for RNA-seq, you will want an aligner that is splice aware, meaning that is able to look for reads that are junction reads, which means that have gaps if you align them to the genome due to the existence of introns. So the output from an aligner is a BAM file, and that BAM file contains not only the sequence of nucleotides, but also the genome coordinates, and it also contains some quality base pair of your sequences. Then once you have your BAM files, typically you will want to count reads over features such as genes or transcripts. And you will go through the step of normalization, read count and normalization. This is not as standard, so usually the output of this step depends very much on the software and how you do it, and you can have that output into a text file or an image file or even a PDF. So before you do an RNA-seq experiment, there are certain points that you should bear in mind. So I've separated them into 4 different boxes. So first thing is to decide your library type. So you can choose to have a directional library or not. By directional library, I mean if you want to keep the information of the strand where the read comes from. I would usually recommend you to always choose a directional library. Because if you don't have your strand information, you might be losing reads. So, for example, for higher organisms such as human or mouse, you have situations where you have 2 genes overlapping, but actually in different strands, in opposite strands. And if you have reads that fall on those areas, what will happen is that if you keep the strand information, you're able to know to which gene to assign that read. But if you lose that information, then you have to discard those reads, which would be a pity. So that's one thing. Then you would like to decide which sort of RNA you want to collect. So you can have a total RNA, if you're interested in that, but you should bear in mind that if you take total RNA, 90% of that total RNA will be ribosomal RNA, and for most studies, you're not interested in that, so you would be drowning up the reads you're interested in. So I would probably try to get rid of that, and you can do that typically in 2 different ways. You can select for mRNA, losing poly-A tail selection. So there's several libraries that will do that, or by depleting the ribosomal RNA in your library. And you can, there's usually the typical method to do this is a ribo0 method. Then another thing you should think of is the sequencing type. So if you want paired end or single end sequencing, for instance, and the length of the sequencing. So single end or paired end means if you want to sequence only the beginning, only one end of your read, or if you want to sequence both ends of your reads. So you can, for a canonical, so if you're only looking for expression of known genes, you can very easily get away with 50 base pairs single end. However, if you want to do something a bit more complex, so if you're looking for, like, for example, splice junction usage, you will need 100 base pairs single end minimum. If you want to do something like novel transcript discovery, you would want 100 base pair paired end, or even 150 base pair paired end, because you would like to have as much information about your reads as possible. That takes us as well to coverage, so number of reads. So I typically recommend for a genome of the size of human or mouse 20 million reads minimum. But bear in mind that if you want to do something like de novo discovery or lowly expressed transcripts, you need much more than that, 20 million at least, 20 million reads at least. Then lastly, but not least, so I would say that you need to think very carefully of how many replicates you want to have. So there's no need for technical replicates. Actually, if you have technical replicates, what you will end up doing in the analysis is that you're going to pull together all the data. So you'll treat it as one single biological replicate. However, biological replicates, you do need as many as you can get. I would say 3 biological replicates minimum, but if you're testing multiple conditions, like for example, if you're testing more than 1 mutant strain or more than one drug or something like that, you would want 4 or more biological replicates. And this is to bear, and you have to also to bear in mind that you should plan for at least one of your samples to fail. Because unfortunately that happens, and then if that happens, if you have a large number of replicates, you're safe. Another thing you should think about is to always randomize across sample groups. So let's imagine that you have 4 replicates for 4 different conditions, and you need to split them across 2 days. You would ideally have 2 replicates for all 4 conditions in day 1 and 2 other replicates of all 4 conditions in day 2. What you wouldn't want to have is for example, all 4 replicates for 2 conditions on one day and all 4 replicates of the other 2 conditions on the other day, because that is a potential source of bias, of adding batch effects to your data. So you would like to avoid that. So this being said, you can perform your RNA-seq experiment, and then you get your FastQ files. And at this stage, you can already do a bit of quality control. You can already check the quality of the FastQ files and see if there's any point in proceeding into further processing of your data. So I would recommend to use FastQC software to do this. It's a very user-friendly software that will provide you with an HTML web page with several plots that give you different information about your FastQ file, and that will help you diagnose any abnormalities in your data. I will show you 3 plots that I took from a FastQC report that I think are very important to look at for an RNA-seq experiment. There are other plots, and I would recommend you to go to the FastQC website to check on them if you're curious about them. So the first one that I want to show you is the per base sequence quality. So this is going to so on the x-axis, you will have the sequence of your reads, and you can see here you have reads of 150 base pairs. And on the y-axis, you have the quality Phred score. So the quality Phred score is actually just the -10 log 10 of the probability of having called a given base incorrectly. So actually, higher the Phred score, less likely it is that you made a mistake. So ideally, you would want a Phred score that is 30 or above, because it means that you have 1 in 1,000 chances of getting it wrong. So you like the green area basically. The yellow area is also okay. You should be really concerned if you get something in the red area. Also, another thing I should mention is that when you look at this, you'll see that the quality is quite okay and in the beginning, and then as you go to the end of your read, the quality starts dropping. And for this read, it's not too bad because it's still within the yellow area. It can be worse. And this is typical. This happens for longer reads, so that's something you should bear in mind when you choose longer reads. Also, there are softwares such as the one I'm going to show you, a trim galore, that will trim any poor quality base pairs you have at the end of your read, so you don't need to worry about it. If you use that software, you don't need to worry about it. If you use that software, you do need to worry about it if you don't trim your read. So this is one plot. Then another plot I would like you to look at is this per base sequence content. So again, on the x-axis you have your reads, so with 150 base pairs, and then on the x-axis you have basically the proportion of the different nucleotides T, C, A, and G. Ideally, you would like something around this area where you have an evenly distribution between T, C, A, and G, around 25% of each. However, for RNA-seq data, you tend to see this sort of bias in the first 10 to 15 base pairs, which is quite unfortunate, but it's a known bias that happens because of one of the steps in the protocol, which you're going to ligate, you're going to prime it with random examiners, and it seems that those examiners are not as random as one would like, so that will introduce this small bias here. However, this being said, it will not really affect your results. So if you see this, you just think, well, it's unfortunate, but that's to be expected. Now, the last quality control plot I would like to show you has to do with duplication levels. And this is a plot is a bit different. So on the x-axis, you actually have the duplication level. So if you have 1, it means that the number of reads that are not duplicated. So ideally, you would have up here at 100. I say ideally because you don't really get that. The blue curve is the one you're interested in, which is the total sequences, the raw sequences. The red curve is if you were to perform deduplication on your FastQ file. But for quality control purposes, you want to look at the total number of sequences. Now, I want to show you this because, again, for RNA-seq data, it's quite particular because you would be expecting a small degree of duplication. And that is because, well, you are probing the transcriptome rather than the genome. So actually the probability of having 2 reads that are exactly the same is much higher than if you were looking at the genome. So actually it might not come from a technical bias. It might be actually that they're real reads, though they look duplicated. Also, you have to bear in mind that for highly expressed genes, the number of those real duplications is higher. And that can be even exacerbated with your coverage, because you will want to recover lowly expressed reads. And those lowly expressed, to recover those lowly expressed reads, you will have to ask for a certain level of coverage, and that will exacerbate the number of duplicated reads you'll get from your highly expressed reads. Highly expressed genes. So this is all to tell you that if you see these little spikes here, that's to be expected. Again, this is typical for RNA-seq data. Other types of genome of data, of sequencing data, might not have this characteristic. So if you see this, you think, okay, it's to be expected. However, if you see a peak here above 60 or 70%, that is not to be expected. That actually might let you know that you have some sort of contamination in your data in your library. And I can tell you that typically that contamination comes from ribosomal RNA. However, you would want to make sure by, for example, taking some of those sequences and running a blast to tell you where actually those sequences come from, to check that it's actually ribosomal RNA. It can be another sort of contaminant. So yeah, that's something to bear in mind. So these are the 3 plots I would like to show you. And so once you've performed the quality control, you are ready to start thinking about processing, especially if you're happy with the quality of your FastQ files. So the first thing you want to do is to match reads to features. Could be genes, could be transcripts. And so the traditional approach is that you will align your reads to the reference genome, allowing for splicing events. And then you will count how many reads map to a set of reference transcripts. And there are several splice-aware aligners, such as star, HiSat2, and top hat. You might notice that I've highlighted star and HiSat2. I've put them in blue. And I've done so because these two softwares have been implemented into Pragee. So if you're running Pragee, you can choose one of the two. Alternatively, you can bypass the alignment step altogether. That's a quite new way of processing RNA-seq data, but it's actually very used already. So to do this, you would have to quantify transcripts directly from the FastQ file. And you have several tools that do this. You have Callisto, Sailfish, and Salmon. So Salmon is the one that I've implemented in Pragee, and so that's the one I'll describe a bit further. So Salmon doesn't do an alignment because it does a quasi-mapping step. And that is done by finding shared substrings between a read and the transcriptome. So in this situation, Salmon doesn't use the genome as a reference, but the transcriptome as a reference. And then it will assign reads to transcripts based on chains of maximal exact matches, meaning it will match those substrings and to transcripts, and it will try to find where would it be more likely for it to be assigned to. So advantages and disadvantages of Salmon. So Salmon is much faster because an alignment usually is the slowest, is the bottleneck, is the slowest step in the whole processing. So if you bypass that, the processing becomes quite fast. Then you have, you can also say that you have it automatically discriminates between isoforms, again, because you look at the transcriptome. However, the things you have to bear in mind is that all reads that are outside of that transcriptome will get discarded. And so with Salmon, it's impossible to do any de novo transcript analysis. Sorry, de novo assembly of transcripts, that's what I meant. But if you don't want to do any of those things, if you're only interested in transcripts or genes that are well known, then I would definitely recommend you to use Salmon because the results are pretty good and it's very fast. So you could get your results, I don't know, but perhaps in a couple of hours, depending on how large are your FastQ files. So once you've matched your reads to features, you can start thinking of expression levels. So counting your reads and normalizing them. Bear in mind that raw read counts are non-informative. They have different sources of bias, such as gene length, GC content, and sequencing depth. Actually, it's quite intuitive. If you think that you have a long read compared to a short read, the long read will have more raw read counts than a short read. And that doesn't mean that actually it's more expressed. So this is one of the things you need to think about it if you want to compare things. So there are several methods to normalize, and I'm just showing you 3 of them. So reads per kilobase million, RPKMs, or fragments per kilobase million, or transcripts per kilobase million. And actually RPKMs are not so used anymore because they instead of that, people tend to use more fragments per kilobase million because it looks at read pairs instead of just reads. But actually the one from all these 3 metrics, the one that is mostly used these days is TPMs, transcripts per million. And the reason is because with TPMs, you can compare across samples. So with RPKMs and FPKMs, you could compare gene in sample X, gene A versus gene B. You could say, okay, gene A seems to have a higher level of expression than gene B. But you wouldn't be able to compare gene A in sample X with gene A in sample Y because it didn't normalize properly in terms of library size. But you can do so with TPMs. So, so usually I would recommend you, if you want to get a metric, to try to get your data into TPMs. And I'm happy to say that that's one of the outputs of Pragee. So if you run Pragee, you would have that already automatically. So once you have expression levels, the logical next question is, do you have genes that are differentially expressed? So by differentially expressed, I'll just give you a quick example. So imagine you have two conditions. You have plus normoxia and plus hypoxia. And you're looking at this gene, HIF1A, and you want to see if HIF1A is more expressed in the plus hypoxia compared to the plus normoxia compared to the plus hypoxia, to see if these levels are actually statistically significantly different. So there are different methods that have been generated to do this, and the most common ones are, well, some of the most common ones are D-seq2, cufflinks, EDGE-R, and LIMA. And I'm happy to tell you that the results across these methods are generally very consistent, so it doesn't really matter too much which method you'll choose. I have incorporated into Pragee D-seq2 and cufflinks. And so D-seq2, it's usually the best method if you have experiments with complex design, such as you're comparing different mutants with different drugs, for example. It will allow you to separate the different factors, like such as with drug, without a drug, or which mutant you want to look at. However, cufflinks is the only tool installed in Pragee that will do you a de novo transcript assembly. So if you're looking for novel transcripts and you're using Pragee, I would definitely recommend you to use cufflinks. So, so I've shown you the three basic steps of the processing, which is matching reads to features, computing expression levels, and performing a differential gene expression. So at this stage, I think that I'm ready to start introducing you to Pragee, which is, as I said, the pipelining software tool that I've developed to process RNA-seq data, and it stands for Pipeline for RNA-seq Analysis with GUI. And I'm happy to say that the software is available online on our GitHub page. So RNA-seq data processing. So it usually goes through 5 steps: quality control, trimming of your reads for any adaptive contamination or poor quality reads, alignment, quantitation, and statistical analysis. So Pragee has all of them. So, and this is the workflow. It's a bit complex, so I hope you can bear with me. So you have FastQ files, and then through Pragee they'll go through trim galore, which will trim your reads, and then trim galore will go through a step, will run FastQC to check the quality of the trimmed FastQs. Then you will have a choice of running a step where you have alignment of your reads or not, or you can bypass it. So let's think that you do want to align your reads to the genome. So you then have a choice of 2 aligners, star or HiSat2, that will then align your reads and provide you with BAM files. For star and HiSat2, you'll need to provide a genome FASTA file. Now, I'm happy to say that if you're running Pragee on the cluster, as I will show you how to do in the practical, Alistair and we have made available genome FASTA files for some of the most common model organisms, such as human, mouse, C. elegans, etc. So it's easier for you to provide those. And I will show you in the practical where they're stored, so don't worry about it. Then so you get your BAM files, and then you have another choice. So you can either choose to analyze the data through cufflinks or through what we call the D-seq branch. So if you go through cufflinks, so cufflinks is not only a tool, a software tool, but it's also a suite of different tools. So the first tool that is called cufflinks, it's a bit confusing, I know. So it will generate assemblies, so transcriptome assemblies for each BAM, and then through cuff merge, it will combine those assemblies, and it will do the quantification with cuff quant. Then you can do cuff norm if you want a specific normalization, or you can go to cuff diff, which does not only normalization, but also differential expression analysis. And cuff diff will then be the tool that will give you the differential expression analysis, and that will give you the file that is compatible with Alistair software, PEAT. And I will show you which file that is in the practical. So don't worry, it's going to be clear for you. Then Pragee will then get the output from cuffdiff, and in R script where it calls the library CumRBund, it will then plot some quality control plots that will help you assess the quality of your sample, well, of your run. So it will run a PCA plot, a dendrogram, and a scatter plot, and that will give it's just a PDF file. Again, I'm going to show you this in the practical. So this is if you choose the cufflinks branch. Now, if you choose going through DSeq and HTSeq, so HTSeq will do the read counting over features. Oh, and I forgot to mention, for both cufflinks and HTSeq, you need to provide a GTF file. But then again, on the cluster, where you find the FASTA files, you'll also find the GTF files. So that can help you. Otherwise, you can find both online. Then, so the HTSeq will do the read counting for you, but it will provide you with raw read counts at this stage. And so then those raw read counts will go into an R script that I've written that incorporates 3 steps, which is an exploratory analysis, which will give you, again, quality control plots. It will do normalization. You'll get TPMs out of it, and you will get the differential expression analysis through DSeq2. And the output from DSeq2 is the one that will be compatible with PEAT. So yeah, now again, I'll show this during our practical. So this is the results if you do an alignment. Now, if you bypass the alignment step using Salmon, so Salmon will match your reads with your transcripts. For that, it will need the transcriptome FASTA file, which is also on the cluster, and it will need a GTF because it needs the information of the features. And then the output is actually compatible with the R script that I had designed for our HTSeq. So the output you'll get from Salmon is pretty much the same as the one you get when you are running through HTSeq and DSeq2. So because yeah, Salmon is compatible with DSeq2. Then finally, because I think quality control is incredibly important, I have added another tool that will provide you further information about your run. So that's called MultiQC. So MultiQC is a wrapper that will provide you a summary quality report in the form of an HTML web page that you can load, and then you can see and you can see the results. And what it does is that it basically looks for the reports of all different software tools, such as HTSeq, STAR, HiSat2, Salmon, FastQC, and it summarizes all of it for all your samples into one single HTML page, which makes it much more tractable to look at and to assess if you have, for example, one sample that doesn't look right compared to the others. So that's definitely something that I recommend you to look at. And it's very, so and the results, I'm going to show you some examples of multi-QC reports during the practical. So yeah, don't worry, you'll be able to have a look at what sort of reports you get from that. So Pragee itself, as the name says, has a GUI. So when you start running Pragee, this is the window that will pop up. Okay, so this is the window that will pop up. And the GUI is divided into 5 sections. So the first section is where you put the information about your input files, so the paths. And the first file that it was going to ask you is a samples file. So the samples file is basically a tab separated file that contains 4 columns. So it contains a column where you'll put your sample IDs, a column where you put the paths for read 1, the column for paths for read 2, and a column that you'll just put which condition each sample belongs to, so that it knows how to group replicates. Then it will ask you for a FASTA file and a GTF file. Then the next box is library information, so which organism, if your library is single end or paired end, and whether it's stranded or not. And for strandedness, you have 3 different options, because you can actually have stranded libraries that are in the reverse strand, so you basically will have to tell the direction. So if reads match the same direction as your genes, you will keep the from start option. If not, keep the from end, you should choose the from end option. And if the library is non-directional, you should just put both, because it means that you have reads that will go through both strands. Then analysis. So these are different parameters. These are different options that have to do with your processing. So the MapQ threshold is a threshold that will be used to select reads. So you can remove reads that map multiply to the genome if you type here the number 20. However, this is also the default, so you don't have to, but perhaps it would be better if you do, so that you're aware you're doing that step. Then you have a read mapping program. So you can choose, as I said, between star, HiSat2, or salmon. So if you press here, you'll see 3 options. The statistical method, either DSeq or cufflinks. If you choose cufflinks, you can choose to tick this box, which will say, please use this GTF file when doing the assembly, so that you don't need to actually redo a total de novo assembly of transcripts. And you can tick this box if you're interested in seeing cuff normal results. So then you have a box for software arguments. So that is because Pragee has been developed in such a way that you can use the default options, which are basically the default options for the different softwares that it uses, the developer's default options. But if the default options are not appropriate for you, for your analysis, you can easily change them by just putting the appropriate options to the appropriate in the appropriate software box. And the way to put those options is the same as if you were running these softwares in the command line. So for example, for trim-galore, imagine you know the sequence of the adapter you want trim-galore to trim. So what you would have to type here would be -A, and then the nucleotide sequence of that adapter, because that's how trim-galore look. That's the flag for trim-galore, for finding adapters. So that's just an example. And you have here all the different softwares that you can, that are used. Now, index folder, it's because Pragee, to do the alignment, will need to generate a genome index if that's not already generated. If it is already generated, you can bypass that step by just telling it where the index folder is. Also, if you want to generate a new index, again, you can put the options you want if they're not the default options here in this box. Then finally, you have process options. which is just like if you want to skip certain steps like FastQC and multi-QC, which I would advise you against, but you could do that. You can, then there's this option here, and please note that this is the default, so it's ticked by default, that is log output to file. So I recommend for you to always keep that ticked, because, well, log files are incredibly important. So for example, if you're ready to publish and you need to fill in your material and methods section, and you need to put which softwares were used, which parameters were used, well, your log file contains all that information. Also, if for some reason your run crashed, the log file will be able, hopefully will be able to tell you where it crashed and what was the error, so that you can solve it, you can rerun it and correct any problems. Now, the quiet box is if you want to keep your terminal clean, meaning if you don't want any messages being warning messages or status messages to be printed onto your terminal. So if you don't want that, you can just type here quiet. Then this box here is very important, submit job to LMB cluster. So if you want to, if you're running this from the cluster, you need to tick this box. Yeah, that's the box that we'll be ticking when we'll be running our practical. So I'll show you how to do that. Then number of CPUs. You can specify the number of CPUs you want to use. If you do not specify that number and you're running Pragee on your local machine, it will just default to the maximum number of CPUs that the machine has, which you might not want to. So you might want to reduce that number. If you're running it through the LMB cluster, it will by default only use 4 cores. But you can increase that easily by typing a different number. And if you tick this box, you can even request a whole node if you have very large FastQ files. Then once this is all correctly filled in, you can press submit, and it will start running your job, and you'll get a progress bar. And you'll see all that in the practical. So another thing is that before you go to the practical, you can get more details about Pragee on our webpage. And I would recommend you to read this Word document, Quick Guide to Pragee, before the practical. I think it would be very helpful for you to understand better what you'll need to do during the practical. So yeah, and it's not very big, don't worry. It's supposed to be quick. So once you run Pragee, you will get 3 types of results files: exploratory analysis plots, like I was explaining, the PCA or dendrogram, files with read counts, normalized read counts, such as TPMs, and then files with results from the differential expression analysis, so the ones that are actually compatible with PEAT. And don't worry, because I'll show you examples of all these 3 during the practical, so you'll be able to see these into a bit more detail. And so I think at this stage I have finished, and I think that I am ready to then swap with Alistair. Well, he'll be introducing you to PEAT.
Thank you for that, Paola. So, as Paola's covered her PRAGUI workflow, at the end of this, we end up over here at the blue ends with, as she described it, effectively spreadsheets of data. So you've got for each gene, you've got its expression, you've got whether it's differentially expressed, and you could look at this in Excel, and probably quite a number of you have at some point. But this is complicated. This is difficult if you want to do anything complicated, particularly if you wanted to look at more than one comparisons. So you had a wild type and 2 mutants. It rapidly becomes impossible to do this in something like Excel. So I wrote PEAT. Whoops, not moving on. I wrote PEAT in order to allow you to look at your data and explore it to a greater degree. So this is an R program, but when you run it, it runs in your browser. So here you can see it. and this can take data from any species, so it particularly is set up for the 5 species listed here, and it can take the different file formats that you can get out from PRAGUI, either on a per gene level or a per transcript level. And then it can give you the data about this in a number of different ways. And I'm just going to briefly go through what these different ways show you. So first we've got the summary page, and this will show you the number of genes or transcripts in your sample, give you some mean values for the different expression levels and log fold changes seen, and how many genes are significantly differentially expressed. And here, this is just without any filtering, so it's got lots of genes, and the log fold change is at not a number and infinites, because some genes are expressed in one sample and not in the other, and that means it's an infinite fold change. But all of these numbers become more meaningful when you actually do filtering, and we'll come on to how you do that in a moment. And then you have the table view, which is essentially the viewing it as an Excel spreadsheet view. And down here on the left, we have a table columns option where we can actually turn on other columns showing things like the value of the p-score for whether things were differentially expressed, but by default that's turned off because normally it's not something you want to look at. And if you have too many columns, it just gets hard to see anything. Now, throughout PEAT, anytime you have something, for instance, like a gene ID here with a blue background like that, that's a link. So you can click on that to go to the web page for this gene. So you've got the table view, and that's just zooming in on it. And you have the per gene view. So here I can look at a particular gene's expression in all the different samples in my data. So here, wild type, P53 knockout, and Rev1 knockout. And these are kind of basically the bits that you could do in Excel if you really wanted to. So going back to the table, as I said, the blue boxes let you do link through. So here in this case, if it was human one, I might click on one, go to the ensemble page on that gene. Or if I click anywhere not the blue box, I go to the per gene tab for that gene. And the per gene tab, here you'll note that there is a star above the P53, showing that there's a significant difference between the black bar, which happens to be wild type, and the P53 bar. Now you can change which the black bar is at the bottom here, so I can change and look for things that are differentially expressed versus P53, in which case I would probably get an asterisk on the wild type and the Rev1. I can choose to use a log scale for the expression. I can do various things to it, so it gives you a few options on drawing it. And for both any tables you see in this and figures like this, the download data option on the left here will allow you to download the tables or to download the figures as SVGs or scalar vector graphic files, which is the kind that papers want in general. So that's all available from PEAT. So yes, as I said, the asterisk tells you things are significant. So then, as I said before, the summary is more meaningful if you do the filter. So if on the left-hand side here we select filter input, we can then filter in different ways. We can choose which comparison we're looking at. So say here we started off with wild type versus P53, but maybe we wanted wild type versus the Rev1 mutant, or the two mutants versus each other. We can filter to choose to look at things that are differentially expressed only. We can filter for different expression levels, for different fold changes in either direction or both. We can look for things in multiple comparisons. So we can look for things that are significantly differentially expressed between wild type and P53 mutant and between the wild type and the Rev1 mutant, or things that are not found in both with the not in other comparisons box on the left here. And we can also filter to only show genes that are found in a list of genes. And we'll get on to how you make a list of genes in a moment. So this also has the figures tab. So this is a bunch of figures that might be helpful or interesting to you to look at your data that people have requested to be in things. So the first is a volcano plot, which is just showing you how the log fold change and the probability of differential expression are for all the genes in the sample or for all the genes that you filtered down to. So here, for instance, we are say showing all genes, but I could have it doing just the differentially expressed ones, and it would end up just drawing the green and red dots at the top. And there's some options you can choose about what it shows. Then there's a scatter plot where I can look at the wild type expression or versus the P53 expression in this case, but any of your sample comparisons. And here the ones that are red circled are the ones that are differentially expressed. Then you can look at a scatter plot comparing across two different comparisons. So this is looking at the fold change on the x-axis of all the genes in the wild type P53 comparison, and on the y-axis, as selected from this box here, all the genes in the wild type versus Rev1 category. So this can be useful to look at whether you've got genes that are changing in the same way or in opposite ways, whether you've got interaction between Rev1 and P53. And here this is showing with all genes, so there's a lot of noise in the figures, you wouldn't expect there to be much correlation. And up here on the left, we have the R value and the probability showing that it's not really correlating. But once we start filtering that to look at differential expression, you might find that there is a correlation. Then we've got clustering. And here I'm only showing it after I have already filtered the data, because otherwise it's got far too many genes. And the notion on this is that once you've filtered down a bit for the differential expression, you would find genes that are involved in the same pathway clustering together because their log fold change of differential expression was changing in similar ways. And indeed, this is what we see here. So these two genes here are involved in similar processes. Then you can also plot a heat map. And here, rather than it being per comparison, so for instance, just for wild type P53, it's for wild type P53 and Rev1, so all of your conditions. And here again, it's been filtered down for the most differentially expressed genes. And we can see there's some clustering going on in terms of genes that are changing in similar ways. So as I mentioned earlier, we can make gene lists. So here we do this through this left-hand side option called gene lists. So by default, the creating gene list option will create gene list of whatever you've filtered to at the moment. So here, that happens to be an unfiltered selection. So if I create gene list, it would just have all the genes in it, but it might be just say the differentially expressed genes in P53. And we can name them when we create the gene list on the left-hand side here, or in fact, we can change the name later. We can also, using this option here, load gene list, we can load previously created gene lists. And the other three options I'll come on to in a second. So that's just zooming in on that. So then at the top here, if we go to the gene list tab, we see the gene list we've created. And it gives us some information about what filtering was done to create them. And when you save gene list, which is done by selecting one of these and clicking on download select a gene list, it saves this metadata, and that will then be loaded in again if you reload the gene list. So here we can see all the genes was this 60,000, the P53 differentially expressed was 55, and the Rev1 differentially expressed was 41. And if I selected multiple of these, I could then use these compare list options down here on the left to look for genes that were found in all the genes that were found in both lists, just the genes that were found in both lists, or the genes that were found in one list and not in the other. And that's just zooming in on that. If I go on the gene list gene tab, I can select one of my gene lists and see what the genes were in it and what their expressions were in the different ones. So here, expression in wild type, expression in P53, and expression in Rev1. Okay. So as well as these options, we can load annotations about the gene. So here I've gone back to the table and I can add new columns that consist of the brief description of what the gene is and the go term of what the gene is. These are loaded using the intermine framework. So these are loaded for the specific species from, say, mouse mine or human mine or fly mine. Not all of these actually have everything here. For instance, fly mine doesn't have brief description. So if you click on it, it'll chug away loading everything from the internet, and then it won't show anything because there isn't any data. But these options are here. You can also add your own annotation file. So here, if you have a text file that is a tab separated file, so it has effectively the first column as, say, for humans, the ensemble gene ID, and the second column separated by a tab, whatever information you want, you can load it here from add annotations, and it'll add it as an extra column at the end. And it'll do this for up to 10 different files. And this is just showing an example of that, where I've clicked on the add Go terms option, and it's added these extra columns showing the Go ID and what the names of those Go IDs are. So as well as all these options, there's another option that relies on uploading your data to something called gprofiler, which can then look for enrichment of different types. So you want to select your differentially expressed genes first. So here I've gone down to the I can't remember, 55 or something, wasn't it? Genes that were differentially expressed in wild type versus P53. And then I can choose that I want to look for gene ontology enrichment, and I can click here to show the enrichment for this. It takes a little while to calculate, then it will show you what Go terms were enriched in your differentially expressed genes. There are also other options than the gene ontology here. This is just showing you what that looks like first. So you get a table which has the Go IDs, descriptions, and the adjusted p-value for whether they were important or not. So here we were looking at the P53 differentially expressed genes, and the Go terms we come out are all to do with cell death, and in fact cell death regulated by P53, so the kind of things that make sense. There's also down at the bottom of this page, another graph which shows you for the top 10 Go terms, which of the genes that they were found with had that Go term. And this can be important because occasionally you find a bunch of Go terms differentially expressed that are actually for two different sets of genes. So I might have just these Go terms found on these genes and these Go terms found on these genes. And it would be important to know that that was going on. So this is just a kind of sanity check for seeing if that sort of thing's happening. In this case, you can see that pretty much all Go terms are found for pretty much all of the genes. So that's not happening here. And here are the other options, as I said, for looking for enrichment. So you have the gene ontology or Go terms. You have KEGG and reactome, which look for pathways. You have a transfac, which is for transcription factor binding sites, MER-base, which is for microRNAs, COROM, which is for protein complexes, and human phenotype ontology, and there's actually another one which again is human specific, so you won't see if you're using any other species. So gprofiler will also let you find orthologs of your genes, so that's what this ortholog tab does here at the top, and that's pretty much all the features of PEAT rather quickly, but they're also described on the GitHub, and there's a little manual for that. So this PEAT is described to be modular, so it's relatively simple to add new species, new annotations, new enrichments. Basically, if you find a website that does the kind of thing that you're interested in and that describes itself as having an API, it's pretty simple to include it into PEAT. And there's also a single cell version of this for those who are interested. So the files and manuals for PRAGUI and PEAT are found on the GitHub, the LMB-seq GitHub, at these links. I find the easiest way to find them is to Google PRAGUI GitHub, because no one else has called anything PRAGUI, so you can find it very easily. So now, any questions for either me or Paula? Max, if you want to unmute yourself and say, I think we've got a few enough people that we can go away with that.
Hey, that was actually just a positive reaction as an applause. But anyway, I can ask something as well. That would be, so I've recently run my own RNA-seq for the first time, and one issue I found was that I wasn't sure which FASTA file or which genome to assemble to. There's a bunch of assemblies that you could choose from. As a starter, I think it's difficult to like decide what should I go for. It's like a bit random. And the same for the GTF file. Wasn't entirely sure if the one that I was using was the most appropriate or not. So I was wondering if you could maybe include like a suggestion or like a standard that you would suggest for human and mouse and stuff.
Well, for PRAGUI, as Paula mentioned briefly, we have put on the cluster versions for the most commonly used species. To an extent, it doesn't completely matter. If you're getting your versions from either, say, Ensemble or from UCSC, basically you can get pretty much the most up-to-date ones. The most important thing is knowing which version you've done and being consistent in it, because that's something you definitely need to know for your paper. Paola, if you want to add anything.
I would just add that if you're using Ensemble, they have different options. And if you're running an aligner, so if you're running star or HiSat2, so if you do the alignment, I don't know if you remember, you could do like with an alignment step. If you're doing an alignment step, you would usually want to choose the gene or the FASTA file that has a tag that says top level. That's if you go from Ensemble. It's usually the one you want to choose. If you want to go, because that says that it contains all different sequences. It doesn't contain, so you have all the information there. So it can align reads everywhere in the genome. No, so you might want to do that. Then if you're running Salmon, as I said, as I mentioned, you want a transcriptome. So you will want the file, not the genome file, not the top level. You will want the one that has a tag cDNA. So you basically want to, when you're looking at it, when you're looking for choosing a genome FASTA file or a transcriptome FASTA file, when you're downloading it, you should try to find that information, so what it refers to. So Ensemble is very good because it does give you, usually in the same FTP page where you get the FASTA files, you will also get a README file, and that usually contains a description of all the different files you have in. And so just bear in mind that you want, when you're doing an alignment, you want a file with a genomic sequence, typically.
And if you're having any problems, do feel free to email, well, probably me at this point, because Paola's leaving in September, and I'll be happy to help you find the right file. Any other questions?
Hi, Paula, I have a question about the choice of Salmon over Callisto. I was just curious to know why you had chosen Salmon instead of the other one.
Well, yeah, that's a very good question. So I think that the results, from what I could tell, were very similar, and also the way the feed is quite similar. And then I used Salmon because I liked their documentation. I thought that documentation was very user-friendly, and since from what I could see from the literature, the results were quite similar, they're quite consistent, I just went for Salmon. But I think that if you run Callisto, yeah, I don't see why, I don't see why not, really.
I'd add to that they are essentially the same method.
Yeah.
One of them, actually, there's some argument about whether one of them just copied the other one. But I would agree that I use Salmon because it's much more user-friendly than Callisto.
I see. Okay, thanks.
Any other questions? Okay, well, thank you all for coming, and have a good rest of the day.
Yes, so yeah, and I'll send an email around with information about when the recordings are going to be available, and I think that they'll be available by Thursday next week. Okay, so well, thank you very much, and bye.